So welcome everyone. Thanks for joining us to our VICC Virtual Molecular Tumor Board. And it's my great pleasure that we have David Kapper here today on this call. David is a neuropathologist and he studied in Tübingen, I believe, and went to Zurich in between, and he's been in Berlin since 2017 as a professor and is working on DNA methylation-based classification of tumors, and that's what he's going to talk about. And we've been trying to get him onto this call for a while now, and I'm really, really happy that he's finally here. So without further ado, the stage is yours, David.
Okay. Thank you very much, Manuela, for the kind introduction. I'm sorry that it took so long until I actually that we found this common spot. It's a great pleasure for me to give you some insight in what's going on in the field of DNA methylation-based tumor classification. As I was introduced as a neuropathologist, quite a bit of the beginning of the talk will be actually on neuropathology and also where this new field of how we can diagnose cancer has actually developed, and I'll go a bit into the history of this. And in the second third, I will then also talk about other types of cancer where there is already published data, and then we'll also talk a bit about nanopore sequencing, which is kind of one of the future technologies that we are currently working on quite a bit. To start off with, I have to declare some potential competing interests, and I think the most relevant thing is that a patent has been filed by the German Cancer Research Center, because that was also one of the places where I worked actually for almost 10 years before joining the Charité Berlin. This patent is on a method of classifying tumor species by DNA methylation profiling, and quite recently, last year, a company has also been funded, of which I'm a co-founder and also a shareholder. So please keep this in mind. I definitely will try to present this as neutral as possible, but keep in mind that I am also involved in these other activities, and also open for questions if there's something in that direction later on. And I think that we will definitely have quite a few minutes of discussion if there's need for that at the end. Okay, so as I said, we will be starting with brain tumors, and quite a substantial change was seen in the WHO classification. That was kind of the 5th edition of the WHO classification for brain tumors that was released in 2021. Amongst other things like new tumor classes, etc. But one of the most fundamental new things that also differentiate brain tumors from other types of tumors is that classification by DNA methylation profiling has been centrally integrated into the diagnosis of central nervous system tumors. You can see here that one of these classical graphs, the so-called TSNE analysis, actually also made it to the cover, kind of cementing this quite fundamental role here for the brain tumor field. And we we believe currently that brain tumors are just the beginning of this new development, and do foresee also a role for this technique in various other types of cancer. Maybe not all types of cancer, we'll have to see this, but definitely for various types of cancer. So what is DNA methylation? I'm not totally sure what kind of the backgrounds of you people here in this call are. So I'll make this relatively basic. I'm sorry if that is too basic for some of you. But just in principle, what we have, this is a very simplified version, is what we're talking about is methyl groups that attach to the DNA, so-called methylated DNA. And these don't attach at random sites, but they typically associate at cytosins, and not all cytosins can be methylated. It's specifically cytosins that are followed by a guanine. So it's a CPG. I think I can change this to a pointer. Yeah. So it is cytosins that are followed by a guanine, so-called CpGs. The P stands for the phosphate group that is in between, and these can be covalently bound by a cytosine, by a methylation group here. And that has many effects. If this happens, it typically doesn't just happen at one cytosine, but it happens at many 100,000 of these cytosins within the human genome. And one of the very simple things or effects of this is that we have this binding of methyl groups in cytosins that are kind of in the location before a gene, typically in the promoter region. And if a certain threshold of these cytosins is methylated here, then transcription factors cannot bind as easily, and then we have a loss of the protein expression. There's also many other roles of methylation, but this is likely the most fundamental one. And in principle, because every cell of our body requires different genes to be active, we have very different types of DNA methylation profiles throughout these CpGs of the human genome. We have something, I think, like 20 million CpGs available in the human human genome, and you will find a certain pattern of methylated, non-methylated, methylated throughout different cell types of the body. So in principle, if you wanted to do this, if you wanted to have something to classify an astrocyte versus an oligodendrocyte or a gut cell or a skin cell, you could record the DNA methylation of all CpGs within this cell and could then kind of build something like a very simple classifier that could tell you, by just comparing this methylation pattern to the pattern of the second cell, if it's the same cell or if you have different cells. So that would be kind of a principal thing that I think nobody has done yet. And also of many normal cell types, we don't yet have such DNA methylation profiles. One very interesting aspect, though, is that these DNA methylation profiles also seem to be suitable to classify different types of tumors. So this is now three different types of brain tumors: astrocytoma, glioblastoma, medulloblastoma, just shown here as examples. And to be honest, we don't actually know why this works, but various studies have shown that these three tumor types have very, very distinct DNA methylation patterns that kind of are very homogeneous, for example, for every astrocytoma. So even if you have two different patients, so the methylation pattern of the astrocytomas in these two different patients will be very, very much the same, and will be very different to other types of brain tumors or other types of cancers. And we don't yet fully understand why this is the case. This is still quite interesting. We assume something like that tumors will develop from stem cells or certain types of progenitor cells at a certain stage of differentiation. And for some reason, for many of these tumors, the DNA methylation pattern seems to freeze in a way at the stage of this kind of cell of origin, and then remains relatively stable throughout the course of the disease. Meaning also that if we, for example, operate a medulloblastoma 3 times during the course of the disease, then very likely the DNA methylation pattern will not change much. I wouldn't say that it doesn't change at all. This we don't know. There will likely also be some changes, but the overall methylation pattern stays relatively the same and stays very distinct from other types of tumors. One word that has to be said about the central technique, and maybe also why this hit pathology, or neuropathology at first, but now is also slowly kind of creeping into pathology. There was kind of the unusual situation at the beginning in this DNA methylation field that there was almost only one assay widely available throughout the whole planet to record DNA methylation data. That was, at the beginning, the Illumina 450K array, which was then updated to this 850K or EPIC methylation array. And this is an array that has kind of a very, very broad coverage of DNA methylation sites. It doesn't cover all of the 20 million, it just covers 850,000, the current version, but it covers nearly all RefSec annotated genes in some way. And there's quite a lot of benefits about this. So it requires very little material, so just 500 nanograms of DNA, which is approximately 5 sections. And the specific thing is that it also functions without problems from formalin-fixed and paraffin-embedded tissue. That was a big advantage of this technique. I'll go into this on one of the next slides. So it is a highly robust method. We have been using this virtually really thousands of times now and have almost no dropouts. We have very little problems with batch effects from these different arrays. So there's a lot we can actually do and compare this data also with other centers. And it is relatively inexpensive. Of course, it's still quite a bit of money, but compared to many other things that we are using, it's relatively inexpensive. But it has one big disadvantage. It is a bulk analysis, meaning that you really need high tumor cell content for this analysis, because every cell you have in your sample, meaning tumor cells, inflammatory cells, all other cell types here, they all kind of add to the global profile, and it can be quite complex to then actually identify what was now the tumor profile, what was the tumor cells, what is the contribution to this profile by inflammatory cells, etc. So there is some things, and there's also some cases where then this performing this technique is just not feasible. So it all started with medulloblastoma. This is the tumor that I mentioned before, which is a highly aggressive tumor of children in the cerebellum. So and in children, it's a relatively frequent tumor. So children have relatively few cancers, of course, but for children, it is a quite well-known tumor type. And for this tumor type, it had been known for quite a while that by RNA expression profiling that requires fresh or frozen material, it was possible to separate this histologically one diagnosis into 4 different subtypes. And these 4 subtypes of medulloblastoma are color-coded below here in these 4 different colors, done by RNA expression profiling. And what was then principally shown here in this work in 2013 for the first time was that the same groups can also be identified by DNA methylation profiling. This is kind of shown in this heat map at the top here. In red, you have highly methylated CpGs, in blue, low methylated CpGs. And what is shown here is that for each of these expression groups below here, you actually have something that almost looks like a fingerprint of red and blue lines that is quite distinct for these different types. It's not super distinct specifically for these 2 groups, and they are still kind of this group 3 and 4, the most problematic ones for us to separate. But overall, in particular for these first 2 groups, we have a very distinct pattern of blue and red lines kind of that you can see here. These different expression types, they are of very high clinical relevance. So for example, this little blue group here is a very benign group of these highly aggressive medulloblastomas. They almost never get metastasis, and there's a lot of, or there's currently clinical trials going on to actually reduce treatment for these patients. These tumors in red, they are driven by sonic hedgehog activating mutations. So there is a clinical trial going on, at least in Germany and likely also in other places, that tries to use targeted therapies for these patients. So there's quite, and these group 3 are, for example, a highly aggressive subgroup here of medulloblastoma, which very early show metastasis. So there's a quite a big clinical need to actually identify these tumors. And in principle, it would have been possible to do this via these expression groups, but that just never really spread out into the pathological field, because pathologists don't like to use frozen tissue, and because we usually don't get it. We have surgery sending in sometimes from over 100 kilometers away from us, and in most cases the infrastructure is dependent on formalin fixation, meaning that for many of these cases, we just couldn't do it. And the big difference here now, or the big advantage that actually was in us from a pathological view, was that it was demonstrated here that the same can be done, so the same grouping can be done from formalin-fixed paraffin-embedded tissues using DNA methylation profiling. That was a big step forward. There was other steps looking at other types of brain tumors here that showed additional properties of what can be done using DNA methylation profiling. And that is something that is still ongoing here, and it is that it has turned out to be an amazingly good tool to detect new types of cancer. What it's done in principle is that, or what was done here is that a tumor that was called central nervous system primitive neuroectodermal tumor, which already, I would say the name of the tumor already indicates that it is a very primitive tumor, meaning that pathologists will likely not have lots of ways to kind of identify what exact type of cancer this could be. They were, a large cohort of these was brought together. They were DNA methylation profiled. Then these profiles were compared with the profiles of other known CNS tumor entities. And these other CNS tumor entities were used as something like a filter. And this is kind of what this filter looks like. And what we mostly use kind of is tools of so-called dimensionality reduction. So this example here, and that was also what was used back then in 2016, a TSME, which is a certain type of dimensionality reduction is shown, which in principle tries to reduce the massive amount of DNA methylation sites that are available. So a dot in this graph here would be one brain tumor where DNA methylation profiling was performed. And what is shown here is in red is the various reference groups that were kind of added as the filter. And what you can see is that they form little islands. And this is what we very typically see, is that tumors that have a shared diagnosis then would also share their DNA methylation profile and form little groups. What you can see, for example, in this group here is that then when the CNS primitive neuroectodermal tumors are added, is that you will have gray dots that will share the same island as red dots, indicating that these tumors are indeed actually not primitive neuroectodermal tumors, but actually very likely belong to this reference group. And this was also shown back in this Cell paper here, that this is truly the case. But what you can also appreciate is that there's other types that do not show any overlap with the reference cases. And these were then analyzed in highest detail. And indeed, it was possible to demonstrate that for each 4 of these groups, they are actually independently driven cancer types that all had different types of fusion genes that are driving this cancer. And also, if you look at this isolated little group, then also have, in a way, a distinct histological pattern that can be identified. And that was actually kind of the birth of 4 new tumor classes. And all 4 of these have in the meantime now been recognized by the WHO classification as new tumor classes that we can now diagnose. So that was 2 things that were kind of shown. And actually already at the time the methylation data of the medulloblastoma was shown, a bigger project was kind of started. And that was that the idea came up that if it is possible to differentiate medulloblastoma into these 4 different types of tumors, which is not possible by histology, for example, then in principle, it should be possible to do the same with all types of brain tumors. So to have a completely histology independent way of classifying if we had all types of brain tumors represented in one centralized reference cohort. We would have to do methylation profiling of all of these samples and bring them all together and do this in a kind of very consorted way. And this is kind of what was done here in a very long-going project that was then finally published in 2018. What you can see here is a depiction of this central reference cohort that was brought together here. That was, back at that time, 82 tumor classes, so nearly covering the whole WHO classification, which has approximately 90 tumor classes. We were not able to kind of get enough representative samples of all classes back then. And each of these tumors here, over 2,800, received a DNA methylation profile, and every dot kind of is one patient sample that you can see here. And this was in principle the first map of the DNA methylation across different types of brain tumors. And what you can see here is that we have the formation of different islands. For example, we have the various IDH mutant tumors at the top here. We have different types of glioblastomas, for example, over here. And we can also see that other types of tumors that are histologically very, very close together, for example, the medulloblastomas, they actually form at least 4 groups within this class here that can be very far away. So even this is one group of medulloblastoma, this is 2 other groups of medulloblastoma, this is the 4th group of medulloblastoma down here. So what you can appreciate is that histologically they are very, very close together. From the DNA methylation kind of in such a map here, they are forming very, very different groups in different regions of kind of this little map here. And of course, the next step is, if you have such a reference cohort, to use it in diagnostics. And that is then, of course, the realm of machine learning. And I can assume that maybe in the further future, when people look back at, okay, what happened at this time here, is likely that people will maybe not even think about DNA methylation that much anymore, because other techniques could also be used to do this, but that this is actually a big step in the direction of that machine learning algorithms can easily be implemented for diagnostic processes in pathology. And what was also introduced already here is a relatively simple algorithm, a random forest classifier that uses this reference cohort and that then allows within seconds to analyze if a test sample belongs to a class within the reference cohort or is unknown. It gives a score of how exact the match is and will then kind of make an automated methylation class prediction. So such an algorithm can be tested. You can do cross-validation of this data set and can try to calculate the misclassification rate, which is, if you just look at the system, around 1%, which is definitely not perfect, because it shows us that we will, in 1 in 100 cases, we will actually get a misclassification. But the big question was, of course, how does this compare to routine diagnostics of brain tumors in a highly specialized center? This was done for over 1,000 cases, and in principle, what was observed here is that in a certain amount of cases, that was 12% back here, the system tells us it doesn't recognize what this tumor is. So it just says not part of this reference set. And I'll talk about these later. So for the other cases, a high quality score, so a threshold was introduced for these scores that come out of this algorithms was identified. And these scores indicated that the system knows what the diagnosis is. And of course, then these were compared to what had this case been classified by pathology only. And there, for the largest number of these cases, there was a confirmation, so that just the system gave the same result. But for 139 of these cases, indeed, a different diagnosis was handed out by this machine learning classifier. And these were then looked at in very great detail at the German Cancer Research Center. And for the vast majority of these cases, or 129 of these 139, it became very clear that, in fact, the DNA methylation classifier was more correct. And the official diagnosis of these cases was then also changed to this new diagnosis. So that led us to the conclusion that if you use such a system, that you can expect a reclassification of approximately 12% of your tumor diagnosis, which is quite a lot. and has led to various studies that also demonstrated that the misclassification or reclassification rate, at least for brain tumors, will be approximately within this range. One further part of one further nice part of DNA methylation data is that it can be quite easily shared through the internet. So it is considered as non-identifiable because you don't do any sequencing, you don't get any sequencing information of the patient sample that you have in front of you. You only get the information if a cytosine is methylated or not. And this is kind of not patient-specific. So in principle, it will not be possible to identify a patient via this DNA methylation profile. And thus, it is for many, many countries much easier to share this kind of data through the internet. And so a website was set up where you can upload this DNA methylation data. You can choose to donate your data if you want to. And within a few minutes, you will get a free of charge DNA methylation report that will give you a few quality scores. It will give you a prediction, will give you a score of how exact was this prediction, and will in addition calculate things like chromosomal gains and losses, or a so-called copy number profile across this case. And this has been very heavily used. So since this publication, it's now getting close to 140,000 cases that have actually been uploaded to this website here. And it is also by us here at the Charité quite heavily implemented, this algorithm. So now within the WHO classification, there's different roles that DNA methylation kind of has found its way into. One is that two tumor classes that were newly discovered by using DNA methylation profile there have been no other methods developed to identify these tumors besides DNA methylation profile. Still, the WHO decided that these tumors should be included into the new WHO classification. And so there is essential diagnostic criteria. So you have to perform a DNA methylation profile if you want to diagnose one of these specific tumors. And this is, for example, the high-grade astrocytoma with pyloid features. So that's something very specific that makes it necessary to use DNA methylation as a technique if you want to diagnose this type of tumor. That has a quite clear reason. So the histology is relatively non-specific, whereas the DNA methylation pattern is very highly specific and distinct. So this was just quite straightforward, the best method to identify these tumors. A second role that DNA methylation is likely used most if you have an unresolved histology, meaning that you have a tissue that is possibly a tumor. It's not entirely clear. It could maybe also be a reactive change or something that you can then perform a DNA methylation profiling. If this then matches with a specific tumor, then by the WHO, you're allowed to officially make this diagnosis, as long as the histology at the beginning would in principle be compatible with such a diagnosis. What in addition kind of has been going on in parallel, I would say, is that many new brain tumor entities have been identified using this technique. So earlier on, I said that at the German Cancer Research Center, there's the request, if you use the website, you don't have to do it, but there's the request for a data donation. And this, many patients and submitting doctors have done this. So the German Cancer Research Center has now brought together a huge data set of DNA methylation cases that is now a much higher granularity map to the one that you saw before, with many kind of new little groups that are being split up, and many research groups are actually working now on understanding why do we see all these little kind of this separation, for example, of this red little country here, which was just a single group in the earliest versions. And that has led to a substantial number now of new brain tumor classes that have been identified over the last years. Most of these have already been added now to the WHO classification. But also beyond this WHO classification, there is kind of new tumor classes that are being identified. And just here showing one example where, so this unpublished here, where a doctoral student of mine is currently working on, is that we would try to identify little islands that have not previously been defined or have not been identified to belong to tumor classes. We would then identify these two cases, would try to contact the submitting centers. If they are able to submit the material to us, we would then perform additional molecular analysis and histological analysis. This, for example, was something that just stood out as a very distinct little methylation group here. We then looked at a few of these cases histologically, and they also had a very distinct histology. Despite growing kind of centrally within the brain tissue, they actually look very much like a schwannoma, which is usually a peripheral nervous sheath tumor. We analyzed several of these cases, and in RNA sequencing then identified that most of these cases, likely all, actually have fusion genes in a gene called VGLL. And we are quite convinced that we have now identified a very, very rare new brain tumor entity. So that's 14 out of these 95,000 tumors that we actually use to identify this. So it's just 1 in 7,000 brain tumors that then turns out to be one of these. And the nice thing about the machine learning classifying algorithms is that they can very easily be centrally updated, meaning that if the central classifying algorithm in Heidelberg is updated, that everybody who uploads data to such a system will automatically get the same update and will from that moment on be able to diagnose all the newly added tumor classes. And so this has been done. The last update was in May 2023. That is now version 12.8 of the brain tumor classifier. And there has been a substantial increase now of tumor classes that are now reaching the 150 of different tumor classes that can be recognized using the system. And this is currently already quite a bit beyond what the even 2021 WHO classification has as a granularity for identifying different types of brain tumors. DNA methylation has added in various fields, so like identification of new tumor classes, but one thing where it is really doing a tremendous difference is the harmonization of diagnostics. And this was also demonstrated here in this little trial and has also been shown by others. In this little trial here, we extracted centrally at the Charité DNA of 4 very tricky different types of brain tumors. We sent out kind of aliquots of this DNA to 12 different European centers, or European and to Australia. And without them, and the other centers were completely blinded to histology, and they were asked to classify the tumors by their DNA methylation profiling and also to generate DNA copy number reports and kind of report what they see as suspicious. So this is, I will not go into detail here, this is for very rare types of brain tumors, very tricky. And if we would do this as a histological round-robin test, I'm very sure that we would get a lot of different types of diagnosis for these cases. But what came back from these different centers was absolutely amazing, in that every single center was able to classify these tumors correctly. Every of these centers with a massively high correlation to the central coordinating center here in Berlin. So this is one example, just demonstrating how highly standardized it is to classify these tumors and how easy it is nowadays for us, if we use DNA methylation, to, for example, make a statement about if I get a data set from a colleague in Madrid and he asks me, okay, what do you think of this tumor? Then if he sends me this DNA methylation data, and if the DNA methylation data is of good quality, then without any further requirement for me looking at the tissue, I will have a very good chance of making a very distinct diagnostic interpretation of this tissue. So this classification or this laboratory comparison trial was actually done to train centers to become molecular hubs as part of a centralized clinical trial for low-grade brain tumors. There's currently other such trials going on, and there's also now commercial people that offer DNA methylation round-robin tests. So if you have interest, you can also ask a question about that later on. So there's other studies that have shown harmonization of this. This was in particular the so-called molecular neuropathology 2.0 trial, which was published in the middle of last year in Nature Medicine, which was a huge trial of many, many different German centers, where there was kind of a blinded analysis of the DNA methylation profiling in comparison to centralized histopathology that was done in Bonn. And there's lots and lots of data that is kind of in this publication. But in principle, also this publication very clearly showed that DNA methylation profiling is a very strong, or can give a very strong diagnostic direction for a specific case, and in some cases may actually be superior to histopathological assessment alone. I will not go into detail into this, but if you have questions to that, please go ahead. Now coming kind of to the last third, which will be then DNA methylation for the classification of non-CNS tumors. And there have been various lines of development, but I think the two main lines that I would like to just briefly show is that on the one hand, we have organ classifiers or organ group classifiers or tumor group classifiers, as I would call it. So there has been the publication of CNS tumors, which has now steadily been updated. Then there has been a publication on sarcomas. This is also just about to be updated for the first time, kind of to a sarcoma version 2 classifier. And there's been other sets, in particular, we've been working on sinonasal cancer, for example, that have also produced kind of something like an organ classifier, meaning that if you have a tumor in a specific region and you're not sure what it is, you can do a methylation profile and then compare with such a classifier and will get a prediction or the information, no, I don't know this type of class, tumor class. Then there's other classifiers that have more the aim of looking at very specific medical questions. For example, you could have the question of a squamous cell tumor in the lung of a patient that has previously had a squamous cell tumor in the oropharyngeal region. And you would then like to know if this lung tumor is a new tumor or if it is a metastasis of the previously developed tumor. That is a very pathologically a very tricky question, but it has been shown that by using DNA methylation, this can be predicted very robustly. Other things could be if you have metastasis in the liver, if it is kind of an intrahepatic cholangiocarcinoma, or if it is a metastasis of a different tumor. There's been classifiers and also a classifier that tried to look at the site of origin of melanoma. Just showing a kind of one example now of such an organ classifier of the top group. This would be on sinonasal tumors. So that was published in 2022. And in principle is the same principle as done for the brain tumor, that a large cohort of different types of sinonasal tumors were brought together and they were analyzed then in this single analysis. And what you can see here is that most of these will form quite distinct groups, for example, olfactory neuroblastoma. But for one specific group, it was clear that not a single class remains, but that it is split up. And this is SNUC, sinonasal undifferentiated carcinoma. Again, an undifferentiated tumor where already the name indicates that possibly it is very hard for pathologists to really come to a definite diagnosis in these cases. And what was quite clear from the methylation data is that these tumors fall into at least 4 different types of tumors. One of these had an overlap with kind of adenoid cystic carcinomas, and we're now quite convinced that indeed these tumors represent kind of strongly dedifferentiated adenoid cystic carcinomas. But for the other groups, it was less clear what these tumors actually represent. And these were again then worked up in great molecular detail. And what kind of got out of this here, or what you can take out of this, is that one of these groups is actually defined by the presence of an IDH2 mutation, One is very, very strongly enriched for SMARCB1 alterations, in many cases a loss of SMARCB1. And a third group is enriched, but not as strongly enriched for SMARCCA4 alterations, and is also has a high rate of ARID1A mutations. So, and likely, and these tumors in many cases have a substantial neuroendocrine profile or immunohistochemical profile. So we currently assume that quite a few, or that maybe neuroendocrine carcinomas of the sinonasal region actually mostly will fall into this group here of these SMARCCA4 tumors. And also from a clinical perspective, this seems to be of relevance. So it is clear that the clinical course is quite different of these tumors, although one has to say that this is retrospective data from many different centers. So this is definitely just the start of looking at these cases. But even here, it's quite clear that these tumors that have this possibly neuroendocrine type that we call then NEC-like, so neuroendocrine-like tumors, and are enriched for this SMARCA4, that they have a relatively favorable diagnosis, whereas other tumors, in particular the SMARCB1 tumors, have a much worse prognosis or outcome here. And also for this here, a machine learning classifier was generated by now colleagues from the Ludwig Maximilians University in Munich, and is still being hosted there, and can be just tested, and you can upload data for free. Again, it's the same principle. There's a request for data donation, if that is possible, kind of depending on where you're coming. So then the classifiers for more specific medical questions, just one example here would be a classifier that tries to separate intrahepatic cholangiocarcinoma versus a metastasis from a pancreatic adenocarcinoma, which can be quite a tricky diagnostic question. DNA methylation turns out to relatively well separate pancreatic adenocarcinoma here in black from a large part of the intrahepatic cholangiocarcinomas in red, although there is some evidence that there will be further little groups in between. So there's further work that has to be done here, but the main classes are very well separatable from between these two groups here. Then we also looked into kind of further things, and one is, for example, melanoma metastasis and the site of origin prediction. And here we tried to generate a classifier that would allow, if you have in particular a mucosal melanoma or a cutaneous melanoma, to allow us where exactly does this tumor come from, and in particular also for the mucosal melanomas, of which organ origin is this from, because that can be very complex in a clinical setting. A cohort was brought together here. This is also again by Philipp Jurmeister, and as a kind of a surprise, maybe, we did not see any grouping according to location of the primary tumors. So we assume that this has something to do with the cell of origin, that this melanocytes develop in the neural crest. They then kind of reach or migrate into the various organs, and that what we see when we record the methylation profile, a lot of this will be the cell of origin profile. So likely they are just too closely related, these cell types, to really make a distinction and really see distinct classes coming up here. So that does not work, at least not from using the DNA methylation profiling. It may work for uveal melanoma, which seemed to be more distinct to the other tumors, but it didn't work for any of the other types, which was also something interesting and shows us that there will definitely also be limitations to what we can do by DNA methylation profiling. Now, just a few slides on future perspectives, and coming towards the end, is that there is alternative testing methods that are coming to the market. One big thing here and one new star of the show is definitely nanopore sequencing. Nanopore sequencing, as some of you may know, is a kind of a new type of long-range sequencing that has various positive properties for diagnostic implementation, and There has been various studies now looking at nanopore sequencing for DNA methylation profiling, because there is one very big advantage of this sequencing type, and that is that in doing this sequencing, you don't only get the DNA-based information, like in standard Sanger sequencing or in many next-generation sequencing approaches, but in addition to this, you can identify if a cytosine is methylated. You can also identify other of such groups attached to bases, but the most commonly used is actually if a cytosine is methylated. So this can be predicted, meaning that you have suddenly a sequencing method that allows us to do DNA methylation profiling. It also has clearly some disadvantages. For example, we are losing this big benefit that we're able to use formalin-fixed paraffin-embedded tissue, because nanopore sequencing needs high molecular DNA, means that it will require frozen tissue again. And it's not so straightforward to actually use the data as with the standard DNA methylation data from arrays, although quite a lot of these new tools are currently developing, and I think this will not be a long-lasting problem. And you have further advantages, and one is that you have a massive acceleration of data generation. The machines are also very inexpensive, and it could be quite an interesting technique if you have more a low throughput laboratory. Two little examples here on this acceleration is that this technique can be kind of made so fast that you can even attempt an intraoperative DNA methylation classification of brain tumors, meaning that for a standard array-based DNA methylation analysis, we usually take 1 to 2 weeks. That's just the time that these processes take in our laboratory. Using nanopore sequencing, if you just look at a single case, the sequencing goes so rapidly, usually just 20 to 60 minutes here, and the analysis pipeline also is, in many cases, not that long, that in principle, you could have a report almost within the time of the actual surgery, meaning that, and in this report, you can separate out all the different types of brain tumor classes, kind of almost at the same granularity as you can do with standard methylation profiling using an array. That was shown here, and also in this study here, it was attempted to kind of state the impact on surgical strategy, if there was a definite impact of this, yes or no. But I can envision that there could be many other types of cancer where this would also be of big benefit to think about having an intraoperative molecular diagnosis. And here, DNA methylation profiling would be a very good bet. There has been a second group here from Utrecht, from the De Ridder lab, that did not use a random forest-based machine learning, but used a deep learning, so a neural network-based approach using nanopore data, and they were even faster using kind of this, and I think it's something like 40 minutes that they require to come to a diagnosis. So they indeed propagate this as an entire molecular diagnosis during surgery. So we are currently doing first testing also in our in-house samples using this algorithm also, and it does look relatively promising. There's further things that DNA methylation profiling can do, and one thing is that we can also classify cell-free DNA that has been kind of shedded off a tumor, for example, into bodily fluids like the blood, or this is what we are using mostly in neuro-oncology, the cerebrospinal fluid. But you could also imagine other types of fluids like tumors in the gastrointestinal tract that could kind of extract fluids. And so there could be quite a lot of possibilities here. And this is a nice study here from a group in Hamburg, from the Ulrich Schüller lab. What they demonstrated is that you can take a CSF sample of a brain tumor patient. So these were all patients that had a known brain tumor on the MRI imaging. And you can extract DNA, specifically the cell-free DNA. So you're not looking for the DNA within cells, but you look at the freely floating cells, mostly of necrotic tissue that is given over to the CSF and likely enriched in the CSF compartment. And this can be then sequenced using nanopore sequencing, and then they were able to generate copy number profiles of this, but also to use the previously shown DNA methylation-based classifiers, and were able to classify these tumors in a certain number of cases. So for them, they were able to find this free circulating DNA or cell-free DNA in approximately half of these cases, and they were then able to identify or come to a tumor diagnosis again within half of these cases here. And so these were patients that had not been previously operated on, so showing that possibly for some tumors that are, for example, in the deep brain region, or possibly for other reasons that patients don't want to get operated on, there could be a possibility for some of these patients already today to come to a full pathological diagnosis using nanopore sequencing of cell-free DNA. So with that, I would like to come to an end. So I hope I was not far too long, but I do hope that we will still have a bit of time for questions.
Thank you so much. That was a very impressive presentation and a very informative overview. I would have one question right away, and then there are already questions in the chat. I was wondering first, when you started to present, that this would be a great way of classifying and diagnosing CUPs, but apparently that doesn't seem to work that well based on the metastasis result. What are your thoughts on that?
So there have been publications on CUP, so of carcinomas of unknown origin, and this seems to work relatively well. It doesn't seem to work for identifying for a metastatic melanoma. So in these cases, it can just be stated that it's a melanoma, but otherwise there is relatively good functioning classifiers available. We are ourselves working on such a system, which has not been published yet. There's also from Manel Esteller from Barcelona, Spain. He has published a CUP classifier a few years ago. And there's also, I believe, a company and a patent that they have in this field. I cannot tell you the first author, I'm afraid, but Manel Esteller was the last author of that. So there is a great possibility for CUP classification, definitely.
Oh, okay. So that's kind of good news then as well for patients to go forward in that direction. There are questions from the chat. So one question is, please comment on what it means to have such a robust, highly differentiated classification on personalized drug treatments now or in the future.
On what type of personalized drug treatments?
Yeah, so how does that relate then, the diagnosis relate versus personalized drug treatments?
Yeah, so I think in principle, what we are doing at the moment is that we are cleaning up a lot of pathology, and that we are redefining kind of what we see as a diagnostic group. And this, for example, in brain tumors has already reached clinical trials. So there have been some clinical trials that have been stopped because of DNA methylation profiling. For example, of this example that I gave you earlier on of these primitive neuroectodermal tumors, there was actually a long-going clinical trial running, studying these types of tumors. And after DNA methylation reanalysis of this kind of the first part of the clinical trial cohort, it became clear that this is not one tumor entity that people are looking at, but many different types of cancers, and that therefore very likely the results of such a clinical trial would also almost be meaningless. And so the trial was terminated. And the same is happening also now kind of in a prospective way, that for brain cancers, most clinical trials nowadays will start to require DNA methylation profiling to enter a patient into this clinical trial. So what is happening here is that we are seeing a cleaning of clinical trials. And what I showed you about this harmonization across institutions, that will also be the case across clinical trials, meaning that it will be much easier in the future if you have a centralized kind of DNA methylation profile of these tumors to really compare and maybe combine clinical trials after they have been published in a much more straightforward way. And we believe that this is one important thing of kind of another phenomenon that we're currently seeing, which is, of course, that we are seeing kind of new detection of smaller and smaller and smaller tumor classes, where currently we believe that many of these tumors may be amenable for a specific personalized treatment, but they are just far too rare that a center could ever come up with a clinical trial if we don't have such a global system to identify these types of cancer. So there we also think that DNA methylation will play a role for kind of lying in front of or providing a possibility to come up with highly specific personalized trials globally. So maybe that was a bit far-fetched, but that was I hope of some interest, and you can ask an additional question if you have further questions in that direction. Manuela, I cannot hear you.
Yeah. It's definitely an interesting aspect to kind of harmonize also clinical trials. And I see Andrea has a question.
Fantastic talk. Apologies, my camera is not working today. Great talk. I was wondering, and maybe this might reflect on the questions that are in the chat as well. As you said, DNA methylation is usually considered not, sensitive information because it doesn't allow for, or currently we cannot re-identify the patients. But I was still wondering whether maybe in the future we could add sequencing, other type of sequencing data. Have you considered perhaps also using federated learning? So maybe centers in different parts of the world might be more, even further comfortable in sharing data or sharing the models in that case.
Yeah, I think that's a very important aspect. I think that this technique that we're using at the moment of arrays is likely just this is what we're using at the moment. I don't think that in 10 years' time we will still be using arrays because they're just too complex. And I assume that sequencing, either bisulfite-specific sequencing that is also then able to identify DNA methylation or other types of sequencing will pick up. So I think it will not be long until we will have the same problem with DNA methylation data again, that it will be attached to genomic data, so that it will not be able to share. That we will not be able to share as easily as we can today. And this is just because the prices will go down and down for sequencing technologies and likely not for these arrays. So this is at least my assumption here. And then I think you're absolutely right that processes like federated learning will become very, very interesting. There is various projects. I'm also partially involved kind of in one that tries to build such an infrastructure within Germany. But I think that this will likely also be the way for the further future also for DNA methylation.
Thank you.
Yeah, maybe just to pick up on that. So there's one question. So what do you believe how would you think additional data like RNA sequencing mutations and proteomic data might actually change classifications?
Yeah, so I'm just going back a few slides. So that is a very important question because, of course, now I focus on DNA methylation. I'm just going to, to this one here. I just focus on DNA methylation, but that's, of course, only one layer of the information. And it is in a way that, at least for brain tumors, we are seeing a very, very big overlap of driving alterations and DNA methylation classes. We don't fully understand why this is the case, but we see that if a tumor has a certain methylation group, then the chances that we will find, for example, a VGLL fusion within this case is extremely high, often approaches 100%, and we will almost never find such a fusion in a different case. So we are currently using methylation in many of these instances to predict what type of alteration will we detect in this tumor. And this is very beneficial for brain tumors because we have a huge range of different types of cancers, and it's possible that it will also work in other types of tumors. But there will definitely also be further layers. And one thing that I just wanted to point out here, for example, is that we have very distinct looking classes. For example, looking now at these SNAC classes here, But when we did proteomics of the same cases, we suddenly saw something quite different. And that was that this group that lands over here, that was very different from this group over here. By proteomic analysis, they cluster very tightly together and seem from their kind of proteome profile, they don't seem to be very far apart and actually intermix quite strongly. Whereas by DNA methylation, we see this very, very distinct grouping. So this is just one example of where I think, yes, definitely other techniques will add in. And this grouping of DNA methylation is, of course, just one view. And I think for treatment approaches, etc., it will be quite important to also incorporate these other types of kind of technologies.
So one could actually, I now speculate that some of the like smaller groups might actually go back together from the perspective of treatment then.
That could be possible, or that we see a further splitting up of groups. So I wouldn't be too sure in what direction exactly it goes, but yes, both could be possible.
Yeah, okay. And you already hinted that you see specific aspects when you look into the groups. And there's one question, are you also seeing specific features or patterns of the CPGs in the groups, that you have certain patterns in there, how often they come up also?
Well, so is that, so we, of course, definitely we see patterns of the distribution of the CPG methylation across the genome. Otherwise, this is what we use to classify these tumors. And is there, sorry, you may have to ask.
Yeah, so I think the question goes, if you see specific patterns, it might help to develop a tumor type specific approach versus one model for all. So really searching for patterns rather than using a classifier. I believe that might be the question going in.
Okay, that for example, I hope I get this right. So we have for brain tumors, we have the promoter methylation of the MGMT gene, which is a predictive marker for the usage of a specific type of chemotherapy, which is called temozolomide. And of course, we can now, and this is just kind of the promoter of one gene. And what we can now very easily do, and we have actually done this just a few weeks ago, is that we can very easily look at this specific promoter across all types of methylated tumors that we have analyzed in the past. Meaning that coming from this data, we can kind of predict if there are other types of cancer that may also benefit from such a type of specific chemotherapy. Maybe it goes in this direction. So of course we can kind of look a bit more in a holistic way and can try to see broader patterns. But otherwise, I think we would have to discuss this in more detail via mail. It would also be a possibility.
Yeah, I would forward that to the guest who asked that. But I believe, I mean, that's an interesting aspect to say that we basically are stepping away from a tumor entity-based analysis and rather say, okay, let's go for molecular patterns and use that for treatment. And I mean, that might be an approach as well. I have one final, more technical question, which is coming from the audiences. Do you deconvolute the probes, and I believe the ones from your methylation array, to include probes that are somehow more important to tumor cells, to basically enrich for certain areas?
So we don't do this yet, I would say. So at the moment, what is used kind of for most of these classifiers is that there is just a selection for the probes with the highest variance amongst these groups that have been added. And this filtering step of looking at the highest variance of these different groups here will usually give us already a focus of the CPGs that kind of likely, well, are kind of the most distinct among these classes. So there is a selection, but what we don't do, for example, at the moment is that we deconvolute and try to extract inflammatory cells or mesenchymal cells or these kind of things. So that is something that we are working on, and there could be certain entities where this actually improves the classification powers, or where we may actually even then see that if we do this deconvolution, that we are able to identify further types of tumor classes. But it is technically not that simple to then generate a DNA methylation-based classifier that kind of has a prior step of deconvolution. At least we were not able to do this in a clean way so far.
Okay, I hope that answers the question. And we're already over time, but it was so interesting and there were so many questions coming up. Do we have a final question from the audience?
I do have one other question, sorry. Great presentation, Dr. Kapper. Just one question on FFPE specifically. You did mention in your slides that you don't see site-to-site variations when samples are submitted if it's good quality. But what about FFPE effects? Like, the way the sample is prepped in FFPE, is that affecting any of the patterns that you see or your predictability through the classifier?
So there is, in the earliest times, when we started to build this brain tumor classifier, we had problems of probes that we kind of had a grouping by material type, so of frozen tissue versus FFPE tissue in earlier cluster versions. And what was then analyzed is that, or what was then identified is that this actually affects specific sites on the DNA methylation area, and that these can be quite straightforward, be filtered out. So of these kind of 850,000 CPGs that we use, there is a certain number of CPGs that we would not take into consideration because of this problem, that they seem to be more strongly affected by different types of processing. So for routine diagnostics, kind of with using such classifiers that adhere to these things, we see no problem or no differences between frozen tissue and formalin-fixed paraffin-embedded tissue.
That's great to know. Thank you.
Okay.
Okay, great. With that, I would say thank you everyone for joining. Thanks, David, for coming in and giving this great presentation. That was awesome. And I have a lot of questions. I think I'll have to come back to you later.
Yeah, you're welcome. Okay. Thank you very much. Thank you for your interest.
Thanks, everyone.